Biotechnology. So what is biotechnology? The technical definition of biotechnology is the exploitation of biological processes for industrial and other purposes, especially the genetic manipulation of microorganisms for the production of antibiotics, hormones, and etc. Some common applications include medical biotech, agricultural biotech, industrial biotech, and environmental biotech. So, medical biotechnology is one of the most common uses today, and the technical definition would be the use of living cells and cell materials to research and produce pharmaceutical and diagnostic products that help treat and prevent human diseases. An example of this today would be stem cell research. So lately, they found a new type of cell that was made in 2017. This is called extended pluripotent stem cell. I butchered the pronunciation, which is the foundation of every cell in the body. So this is regarded as like a first step to help regrow human cells, replacing limbs and organs, and even healing cancer patients. Some other examples include genetically engineered insulin, which is obviously for diabetic patients, gene therapy, and molecular diagnosis. The next type of biotechnology is agricultural. This is the area of agricultural science involving the use of scientific tools and techniques, including genetic engineering, molecular markers, molecular diagnostics, vaccines, and tissue culture. So to go in more depth, genetic engineering allows for the transfer of useful characteristics into plants, animals, or microorganisms by inserting genes from a different organism. Farmers mostly use this technique on crops to increase productivity by reducing crop damage. Molecular markers, through the examination of DNA, scientists can identify a desirable trait, even if that trait isn't visible, which allows for successful breeding. So tissue culture is the regeneration of plants in the laboratory from disease-free plant parts. This in turn allows for the reproduction of disease-free planting material for crops. So industrial biotech. Industrial biotech is the biological resources for industrial processes, which include pollution preservation, resource conservation, cost reduction. Examples include production of bread, yogurt, and vinegar using microorganisms, and the use of enzymes in detergents. This technique is mostly used by companies and like factories and like cost reduction, so like to maximize production and stuff like that. So the next one is environmental biotech. This is the biotech that is used and applied to study the natural environment. It could also imply that one tried to harness biological processes for commercial uses and exploitation. The goals and examples would be bioremediation, prevention, detection, and monitoring, genetic engineering for sustainable development and better quality of life. This is pretty straightforward. It's not that it's not that too deep, I guess. It's just environment, biotech. So the topics that you'll see in AP Bio include gel electrophoresis and PCR. Gel electrophoresis is the laboratory method used to separate mixtures of DNA, RNA, or proteins according to molecular size. So this is induced by an electrical field through a gel that contains small pores. A PCR, otherwise known as polymerase chain reaction, is a method widely used in molecular biology to make many copies of a specific DNA segment. To go more in depth in gel electrophoresis, so in the gel, like I mentioned before, there's an electrical field. In this electrical field, there's two sides with one positive and one negative. So in terms of DNA and RNA, since they are negatively charged molecules, and we all know that opposites attract, the DNA and RNA are going to go to the positive end of the gel, which is the side for the separation to start. And for proteins, this is different. Since proteins are not negatively charged, researchers must add sodium diadesyl sulfate I don't know how to pronounce that. So basically, this allows for the protein to unfold into a linear shape, and in turn shifts them into a negative charge. So this is a PCR animation just to see show how it works. So notice in the video that the DNA is raised to a temperature of 94 degrees Celsius to 96 degrees Celsius. This basically starts the denaturing process. And after this, the temperature goes back to around 55 degrees Celsius, which allows for the left and right primers to anneal to their complementary sequences. Then the temperature is raised again so Taq polymers can attach to a priming site and extend a new DNA strand. So this concludes cycle 1, and the same process is repeated in cycle 2 and cycle 3. So the questions.